still missing in action, so I'm still Chris today. Today we have the honor of having Dr. Jack Gallant from UC Berkeley here, and I'll have John give the introduction.
Good morning, everyone. My name is Jan Skersetak. I'm postdoctoral researcher with Professor Brown here at UCI, and it's my distinct pleasure to introduce today's speaker, Dr. Jack Gallant, a distinguished neuroscientist and a professor at the University of California at Berkeley. Dr. Gallant holds appointments in the departments of psychology and electrical engineering and computer science. He's also affiliated with programs in neuroscience, vision science, bioengineering, and biophysics. He's renowned for his pioneering work in functional brain mapping and computational modeling, which we'll hear more about today, of how the brain works. His research applies a combination of neurophysiology, functional MRI, and computational modeling to understand how the brain represents information about the visual world. Gallant's work particularly focuses on how these visual representations are modulated by attention, learning, and memory, and providing thereby insights into the complexity of the visual dynamics and visual processes. Dr. Gallant has also developed unique techniques for decoding and reconstructing these visual representations directly from brain activity, and earning him thereby recognition in the field, which is evident by publications in prestigious journals such as Nature, Science, and Neuron. The influence of Dr. Gallant's contributions is further evidenced by more than 20,000 citations and an age index of 58, according to Google Scholar. Professor Gallant's laboratory has created some of the most detailed functional maps of the human brain networks that mediate vision. This work not only enhances our understanding of fundamentals in visual processing, but has also significant implications for both healthy participants and patients' diseased visual systems. So with that said, please join me in welcoming Dr. Jack Gallant.
Thanks very much for that nice introduction. I'm happy to be here. I usually am asleep at 8:30 in the morning, so I'm a little slow. I've got this very large coffee. Hopefully that will solve the problem. My lab does MRI these days. We do a lot of basic MRI, and now in the last 5 years, we've been trying to move our very kind of computationally complicated MRI paradigms into the clinic, and I'm optimistic that it's actually going to work in the next few years. Right now, clinical fMRI is not used at all for clinical diagnosis, prognosis, and monitoring, and I find that kind of annoying. So we're trying to fix that. But I'm not going to be talking about that today. Today I'm going to be talking about an older project that goes back to our old physiology days. I have long been interested in and focused on the early visual system and the intermediate visual system, in particular a visual area called Area V4. Some of you have probably seen this in a textbook somewhere. This is a flat map of the macaque brain. And the macaque brain is about 50% visual. Your brain's about 25% devoted to vision. Because, of course, you have a huge expansion of your prefrontal cortex. And the macaque visual system is divided into something, the low estimate is probably something like 30 visual areas. But since it's difficult to determine the identity of visual areas if you don't have retinotopic maps, and the high-level visual areas are not well retotopically mapped, there are probably something more like, say, 40 to 50 visual areas in the macaque, probably more than that in your brain. And these visual areas are organized into a hierarchy from early vision, of course, the retina, and then the thalamus, and then primary visual cortex, area V1, which comprises about 10% of your brain. It's the largest single area in your entire brain. And then from there, information flows into a complicated network of different visual areas with extensive crosstalk between the areas and extensive feed forward and feedback interactions between these areas. Traditionally, you guys all heard this in some textbook. The visual system is divided into a what stream, which is involved in object recognition, and a where stream, which is involved in the coordinate transformations you need to implement eye-hand coordination and to get around in the world. But the true answer is a bit more complicated than that. And of course, the more we learn about the true underlying anatomy of the brain, the more complicated this wiring diagram gets. But the basic division of what versus where stream is a reasonable kind of first approximation to think about how vision works. And the area V4 is essentially an intermediate vision area on the what pathway. It's an intermediate area that's involved in object recognition. When I started recording from area V4, which was about 1,000 years ago now, V4 was kind of considered a relatively high-level visual area. And the summary of this talk is going to be actually V4 is as dumb as a bag of rocks. It's pretty much a low-level visual area, and that's where we're going to go with this talk. V4, interestingly, even though it's only 2 synapses up from V1, right? We have information goes into primary visual cortex V1, goes into secondary visual area called area V2, and then in the cat, these would be area 17 and area 18. And then it goes from area V2 into area V4. And so that's only a couple synapses up from primary visual cortex. So the question has always been, what happens in those synapses? Of course, every time you go from one area to another, there's a nonlinear transformation because neural responses are rectified and summed and pooled. So you have 2 levels of nonlinear transformations from primary visual cortex, an area that we know well. And the question has always been, what are the neurons in V4 doing? And that's today's talk. Summarizes our long period of research on this, and I think it gives us a really good model of area V4 that has a really interesting take-home message, which I'm going to lead with here. V4 is basically implementing V1 on the outputs of V2, and V2 is implementing V1 on the outputs of V1. Basically, the whole early vision stack appears to me something like an area V1 that's just iterated over and over again. And because there's nonlinear transformations and pooling between V1 and V2 and V2 and V4, even though these higher order visual areas are implementing the same computational processes on their inputs, the results end up being different. So how do we know that V4 is a hierarchical, excuse me, try this again. So one of the interesting things about vision is that it exploits the hierarchical statistics of the world. So, the world is made of pixels, but these pixels are obviously not random. If all of the pixels in the world were random, the world would look like white noise. And the only thing that looks like white noise is white noise. So the world has structure in it, and that structure consists of essentially a stack of increasingly complicated statistics. We have the power spectrum of the image. We have the 2-point statistics, the 3-point statistics, the 4-point statistics. These are essentially the higher order correlations between pixels or pieces of an image. And natural images are built of these statistical relationships. And these statistical relationships emerge because natural images are usually opaque. So the appropriate statistical model of natural images is something like a falling leaves model, where objects near you are occluding objects behind you. Objects near to you are occluding objects far from you. And all of these occlusions get piled up on top of one another. And of course, you can see any image, in theory, from any point in space. So when you have a sort of falling leaves occlusion model and an observer that can be at any location in space, that creates very, very specific statistical relationships in the signals out in the world. And your visual system, being incredibly highly evolved to process natural images because you're a primate, essentially incorporates those statistics and exploits them and builds them into the very computational structure of your brain. Another system that does the same thing is a convolutional neural network, which is the sort of the first kind of neural network that broke AI open back in 2011, 2012. A convolutional neural network implements very simple convolutional operations, and it just iterates those across a bunch of levels of a hierarchy with nonlinearities and pooling in between the levels of the hierarchy. And you can build a convolutional neural network and show it natural images. And then you can take the features learned by that convolutional neural network and use them as features for regression onto the brain. and you can identify which brain areas are related to which layers of the convolutional network. And that's shown down here on the slide at the bottom right here, on the panel at the bottom right. The early layers of the convolutional network, which are representing the low order statistics of images, are shown in blue in this map, and the high levels of the convolutional network are shown in red in this map. Probably a lot of you haven't seen this map on the right here, so I'm just going to reference very quickly the map on the left. The map on the left shows the back of the brain in an inflated brain. Of course, the occipital cortex is the back of the head, but this is kind of an inconvenient map to look at. So we can make a few slices in it and flatten out your brain like a pizza, and that's what's shown here on the right. So primary visual cortex is on the middle here, and higher order visual cortex is essentially a ring around retinotopic visual cortex, and that's shown in red. So this just shows that in humans, the representation of the statistical information in images is hierarchically organized, where primary visual cortex represents the low order statistics of images, and higher order visual cortex represents the complicated higher order statistics of images. This is also true in our primate cousins, of course. In area V4, which is kind of in the intermediate region of this slide, it represents intermediate statistical properties of objects. They're more complicated than lines, the statistics that are represented in V4, but they're not as complicated as objects. So the question is, what are those sort of statistics and how are they represented in the brain? And many, many, many years ago, hard to believe I'm that old now, I published this paper showing that area V4 neurons are very highly selective for these non-Cartesian gratings that are modulated not in sort of standard Cartesian coordinates like a Gabor function or a sine wave, but they're either modulated in polar or hyperbolic coordinates. And this is kind of the first clue we had when this paper came out that area V4 is doing something more complicated than area V1. Before this paper, people had used the same stimuli that were effective in V1, bars and gratings, standard sine wave gratings, to probe V2 and V4 and MT. And kind of after this paper, people started using more complicated stimuli to try to probe this area. So a lot of the work that's been done subsequently then is from a colleague of mine, Charles Connor, and his student, Anita Pasupathy, who worked to try to understand what features of objects were coded in V4. We know that entire objects aren't explicitly represented in V4, but it was thought that parts of objects would be. So they showed that V4 neurons are selective for little convex protuberances and corners and little shape elements, especially if those shape elements had curvature in them, and that individual neurons in V4 were tuned for these different shape elements. I'm going to skip this slide because it's hideously complicated. Okay, V4 neurons also have other interesting properties. They have some amount of position invariance. So those of you who took a neurophysiology class on the primary visual cortex V1, you'll remember that there are simple cells and complex cells. And simple cells are sensitive to phase of a grating, and complex cells don't care about the phase of the grating. You can think about those complex cells as having some moderate degree of position invariance in the sense that they don't care exactly where a bar is or a grating in the receptive field. They'll respond to any bar or grating of the right spatial frequency content. This position invariance property increases across the visual hierarchy. So in primary visual cortex, in area V4, what you see is that the tuning of a V4 neuron for, say, this polar grating is constant, regardless of exactly where that grating is placed in the receptive field of the neuron. So the absolute response of the neuron may change depending on where the grating is placed, but the relative response to that grating versus other gratings is consistent. So V4 has moderate position invariance, and in fact, you see this position invariance escalate dramatically at subsequent levels of visual processing, so that when you get to the highest levels of vision, those neurons are almost completely positionally invariant, and they don't have a retinotopic map anymore. And this is, reflects essentially the transformation in the visual system from a retinotopic coordinate system, which is really based on a camera that's sticking in the front of your head, to an object-centered coordinate system, which is the coordinate system you need to make judgments about the world and make decisions about the world and to interpret scenes. It's just another slide showing the same thing. So if you are recording from area V4 in an animal, your first question is always, well, what would happen if I had a human with a V4 lesion? Would that human look like an animal with a V4 lesion? And the answer is yes. About, how long ago was this? Wow, 25 years ago now. We had a patient that Bob Knight at UC Berkeley identified. The patient had diabetes, which causes micro strokes. And this person ended up with a micro stroke in the upper left quadrant representation of the visual field only in V4. It was incredibly, the smallest V4 lesion I've ever seen. And so we had the opportunity to study this patient. He was actually identified because he had achromatopsia in the upper left quadrant. And the classic view of V4, although it's wrong, is that V4 is a color area that only processes color information. So when a neurologist sees somebody with a color deficit, they immediately think that it's perhaps in V4. So we studied this person extensively, and what we found is for every test we could give this patient, they looked exactly like what you would expect if you had a primate, a macaque, with a V4 lesion. They could do luminance discrimination in the upper left quadrant, no problem. They could do orientation discrimination without an issue. They could do motion discrimination. That wasn't a problem either. They couldn't see illusory contours, and they couldn't discriminate curvature. But those processes of illusory contour and curvature were intact in the 3 intact quadrants. So that told us that, okay, human V4 is also processing curvature information, kind of like our primate cousin, which is, of course, what you expect if you believe in evolution. All right, so there have been dozens, maybe even at this point, hundreds of studies of shape processing in V4, but we do not have a computational model of V4. We have, well, I shouldn't say that. We have computational models of V4. My lab has produced models. Ed Connor's lab has produced models. They're just hideously poor models. And the reason we know they're bad models is they don't predict well. So if I record from a neuron and I take a training set of data and I fit one of these models with a few free parameters to that data, and then I try to predict responses in a new data set, the predictions are far below the theoretical noise ceiling predictions. We can account for maybe about 20% of the variance that we should be able to account for if the model is correct, which tells us that these models are just wrong in some fundamental way, or they're very, very incomplete. So since I'm, spent many, many years working on V4, I found this frustrating. I am not happy with a statistically significant result. I want a model that accurately predicts the responses of my neuron under any situation that could ever occur, and that explains all the potentially explainable variants. That's when I'm happy. So as you can tell, I'm rarely happy, but I want to be on the vector of getting happy, right? We want to get the best possible model we can get. So we identified 2 potential problems with the previous data that had been collected. One was that people had used very, very simple stimuli to try to characterize V4. V4 neurons are pretty complicated. And if you use a limited stimulus set, then you can only build a model within that domain of stimuli. And then when you try to predict V4 responses to, say, natural images, which is, of course, the ultimate goal, those models fail because you don't have enough stimuli in your set. The second problem we realized is that V4 neurons are way more complicated than one might intuit, and that to get enough data to fit a real V4 model would require far longer neurophysiological recordings than people had collected before. So we started a project by a wonderful graduate student of mine who got rich on Bitcoin and is now no longer in science, Mike Oliver. And Mike Oliver started a series of experiments. He graduated. Then I had a postdoc, Mike Eichenberg, come in and pick up the project, and he did a lot of computational work. Then I had another graduate student come in, Michelle Winter, and she really did a huge amount of work on the modeling end of things and on the cleaning up the data analysis. And then we also had work from a brilliant postdoc of mine, Tom Dupree-Latour, who also decided he wants to get rich and now works at OpenAI. In the Bay Area, nobody stays in graduate school anymore. Everyone goes into tech industry. It's sad but true. All right, so we have 2 problems. One is we want to use complicated stimuli, not simple stimuli. So instead of using little shape elements or Gabor gratings or polar gratings, we just want to show animals' natural movements. So in this experiment, we just went out and got a bunch of nature videos, and those are going to be the stimuli for our experiment. the other parts of this graph are showing a pre-processing step. You guys all know that V1 does not represent information in the Cartesian frame of reference that you see in a photograph. Instead, it performs a log polar transform on the data, expanding the fovea and compressing the periphery. So at the front end of this model, we just kind of hard-coded in the log polar transform. The input to our model is actually going to be log polar transform. That just saves a lot of free parameters that we don't have to fit in our model. Turns out that's important because if you think about, say, primary visual cortex, which is shown down here on the lower right, this is a log polar map. And if you think about a neuron, say in V2 or V4, receiving input from V1, and if it has a Gaussian or circular shaped receptive field, the receptive field of that neuron is also not going to be circular. It's going to be distorted by the log polar map. So neurons in V2 and V4 and all higher order visual areas that have a red topic map, they tend to be elongated due to this log polar transformation. And we want to model that well. So we implement this log polar transform at the beginning. So the first problem was we didn't want to use simple stimuli, so we started using natural images. The second problem was that we decided that the data sets that people were collecting were not large enough. If you are collecting data in a neurophysiology experiment, you might be able to show, I don't know, maybe 10 minutes, 20 minutes of stimuli in one day. We wanted a much, much larger stimulus set. So we used the Utah arrays for now NeuroPixels was the best technology. At the time, these experiments were done, Utah arrays was the best technology. We put Utah arrays into area V4, and we recorded for months from single neurons. Now, if you record from a single neuron over months, what you'll find is that the waveform of that neuron will change on your electrode. That's shown on the upper left here. These are different days, and it's shown in these different colors. And the reason the waveform is changing is because the electrode is actually moving in the brain, or you're getting gliosis around the electrode tip, and so the conditions of recording change. So because of that, most researchers never combine data across days because they're worried that they have different electrodes on they have different neurons on the electrode tip. So we invented this idea called the fingerprint method. Turns out Jim DiCarlo's lab invented this at about the same time, independently from us. The idea is every single day we would come in and we would show our animals the same, how long was this? I think this was like a 30-second movie. And now, because each V4 neuron is very, very different from every other V4 neuron, we could use the functional fingerprint of the peristimulus time histogram across this complicated movie to classify the neurons. And to show you that that works, the upper right here shows the PSTH in the black dots and the average responses in the lines across multiple days, 48 days. This is a month and a half of recording from a single neuron. So you can see that across days, this neuron does not change. It's always selective for certain aspects. These are natural movies we're showing in this fingerprint, right? So this neuron is selective for certain aspects of these natural movies and not for anything else. To show you that that is unusual, on the bottom, I'm showing you 4 peristimulus time histograms for where we're taking different pairs of neurons. The orange is one neuron and the blue is a different neuron. And you can see that in those cases, they're always distinguishable. So this fingerprint method allowed us to stitch the data across days and combine our neurophysiology data sets to have giant recordings from single neurons. So for many of our neurons, we have over 1 million frames of video for that neuron. And I kind of always had a rule of thumb. If you can't figure out what your neuron is doing after getting 1 million stimulus response pairs, just hang it up and go home. Because by the square root of n rule for data, you're never gonna be able to build a model better than that. Okay. So now we've got these giant data sets using natural images, and we have to process them. At that time, 2010 or so, 2012, the convolutional neural networks were just coming into play. Now these have become completely standard tools in many, many areas of neuroscience. The typical way that these tools are used is you train a neural network on, say, natural images, and then you take the features from that neural network and you regress them onto your neuron. And you basically build a model of your neuron that is based on the features that the artificial neural network learned. We didn't want to do that. And you know, this is a useful thing to do. The Tolius lab at Stanford's done a lot of really great work on this. Using this method, Jim DiCarlo's lab at MIT has used this method. It's a perfectly fine thing to do, but it has problems. First of all, there are no CNNs that are available to us normal scientists that were trained on movies. They're all trained on static images, usually not always black and white images, but certainly static images. The second is, in some sense, if you're really trying to build a quantitative model that you understand, it's unsatisfying to use features from a pre-trained neural network to explain your biological system. We would rather have something that comes more from first principles. So what we decided to do was, rather than going this route of using a pre-trained network to fit our data, we leveraged the framework of neural networks and the way neural networks are fit and the wonderful algorithms for fitting these networks to basically build a model de novo from scratch that was essentially a neural network model, but used elements that we understood. That way the model would be interpretable. So you guys all recall that in primary visual cortex, all the neurons can be well modeled as Gabor functions. These are basically just little Gaussian modulated sine waves, and we've known, since the 70s and the giant fights over whether V1 encodes bars versus gratings, that basically the true answer is both. V1 neurons do respond really well to bars, and they respond well to gratings, and that's because the receptive fields are essentially Gaussian modulated sine waves. And there are two kinds of these: simple cells and complex cells. Simple cells don't care about the phase of the grating. Complex cells care about the phase of the grating. And then there are a group of color cells in V1. They tend to be very, very low spatial frequency because you kind of have two pathways in V1. One goes through the blobs, that's low spatial frequency color preferring cells, and one goes through the interblobs. Those are high frequency orientation preferring cells. So we wanted to build a V4 model that only consisted of these units. And the reason for that is we know that these things exist. We know there's tons of evidence from V1 that this is a really good model of V1. We even have ideas of how you build a neural circuit to actually create this kind of selectivity. So by focusing on this specific sort of type of model, or these specific elements in our model, we can be guaranteed that whatever our V4 model is essentially buildable, that all the elements in that model come from biological things that we already know the system implements. Just want to remind you that there's a reason that primary visual cortex consists of a huge bank of these Gabor filters. And that again goes back to image statistics. Every natural image can be built of Gabor functions. And this is one of my favorite videos from Laurent Perrinet. Basically what we're doing is we have a tiger image here, and what he's doing is he's adding in the largest amplitude and then increasingly smaller amplitude Gabor wavelets. That is a Gabor wavelet decomposition of this image. And you can see that these Gabor wavelets, if you just sum enough of them together, you get an image. And that turns out to be true for all images. And the whole reason your primary visual cortex implements this Gabor wavelet transform is if you're trying to build a representation of natural images that is as sparse as possible and as complete as possible, you cannot do better than this representation. So the idea here is that this representation will also be useful for V2 and V4 subsequent stages of processing. All right, so here's the model. This is a V2. Let's build a V2 model first because it's easy. We start with a natural movie, and we're going to have an input module, and this input module is essentially going to be a model of V1. We've got a bunch of complex cells and a bunch of simple cells, and they're going to get pooled together into by some sort of pooling that we're going to fit to our data, and then that pooling will sum to the output of a V2 neuron, right? Because we've only got one stage of processing here. And of course, a very important thing here is that these V1 outputs are rectified. So there's a nonlinear transformation. If we took a Gabor wavelet pyramid and linearly inserted into another Gabor wavelet pyramid, that would just be a linear superposition, and it could be modeled as one stage. The reason you need a hierarchical model is because there's a nonlinearity between each stage of processing. And in the brain, those nonlinearities, the two most important ones, are halfway rectification and pooling. So this is actually something I want to emphasize here is we're going to build a hierarchical convolutional energy model based entirely of V1 units that is going to look kind of like a convolutional neural network. But if you take a standard convolutional neural network that's trained on natural images like BGG or AlexNet, Those neural networks have millions of parameters. This framework allows us to build a hierarchical neural network model that has very, very low number of parameters. So for this V1 to V2 transformation, we only need to fit 10,000 parameters. In my lab, that's a trivially easy problem. The features of the model are not elements of the stimulus that were learned by some neural network. They're biologically interesting computational subunits that we know exist in primary visual cortex. All aspects of this model can be implemented in known neurobiology. We know the neural circuits that are involved in pooling and Hathaway rectification and representing the Bohr features. So this model is particularly simple and elegant. So does it work in V2? We had a Utah array that overlaps the border of V1 and V2. You can see that border in red here. So remember, a Utah array is just a kind of a bed of nails. It's a 10 by 10 grid. And you can see down below this line, all these neurons look like simple Gabor functions. And above this line, they look somewhat more complicated. They're tuned for color, tuned for orientation. They have larger receptive fields, and it looks like there might be some curvature tuning as well. And that's what you'd expect based on all the physiology we have from V2. I should point out that this array is actually in the fovea. So this also gives us a chance to look at the foveal representation of receptive fields. For those of you who aren't in the physiology business, you might not know it, but almost all neurophysiology in vision comes from the visual periphery, not the fovea, because it's too hard to record from the fovea. And so there's been a long-standing question for like 50 years. What do foveal neurons look like? Are they the same, or is the fovea doing something different? And we can actually answer that question with these data as well. So the short answer on this is this hierarchical convolutional energy model really does a great job of predicting responses, both in area V1 and V2. And you're going to see in these slides something called CC norm. CC norm is the correlation between the predicted responses and the true responses normalized for the noise ceiling. So just a small statistical detour here. When we're doing any experiment in the real world, if we build a model of a fit data set and then test it on a separate test data set, that model can never predict the test data set perfectly. And that's because the fit data set is not the population, it's a sample from the population. So when we fit a model to the fit data set, it has error that has to do with our sampling. And then when we apply this fit data set to a separate test data set, this test data set is also not the population. It's another sample, and it has its own error. So we can't predict perfectly. But it turns out there are theoretical methods you can use to estimate what the noise ceiling is. So you might say, well, okay, the best, in the perfect world, I would predict 100% of the variance of my data, but the noise ceiling says I can only predict 80% of the variance of my data. So when you build a predictive model, you compare the predictions of that model not to perfect predictions, but to the noise ceiling. And that is called the normalized correlation. So I'm fitting to one data set, I'm predicting on another, and I'm comparing the correlation I obtained to this theoretical noise. And you can see that these correlations are really extremely good. We can predict V1 and V2 responses. I wouldn't say perfectly, but close to the noise ceiling. So this is a really spectacularly good model. It works better than any other model of V1 and V4 than V2 than anybody's proposed. And we can also look at the foveal representations in V1 and V2. So these neurons are within 1 to 2 degrees of the fovea. And you can see that they all look kind of like Bohr functions, just like would happen in the periphery. So foveal neurons in V1 and V2 are basically just like peripheral neurons in V1 and V2, which from my point of view is nice because the natural image statistics community has always assumed that the reason you use Gabor functions was, the reason the visual system implemented a system that had Gabor functions as its basis was because those are matching the statistics of natural images. And you wouldn't expect that to change in the fovea versus the periphery. The only thing that changes between the fovea and the periphery with regard to this model is the size of the receptive fields and the spatial resolution. All right. So that's fine. V1, V2, whatever. What we really want to do is build a model of V4. So this is where it gets interesting. Because what we do is we have our input images. We take the log polar transform just to deal with this nonlinear warping of primary visual cortex. Then we have a V1 module. V1 module is just a bunch of simple and complex cells whose output is rectified. And then they're pooled together, and they go into That's called the mixing module in this slide. Then they go into a V2. How is the V2 built? It's a bunch of Gabor wavelet functions. It's a bunch of simple and complex cells, but they operate on the output of V1 that has been rectified and mixed together. And you might say, what does that look like? And the answer is, don't try to think about it too hard. You don't have an intuition for what these things are going to look like. Then we take this V2 representation, and it gets rectified, and those V2 neurons get mixed together and fed into a V4 module, which basically is also just a weighted sum of the outputs from V2. So every V4 neuron, I'm going to fit one neuron at a time here, and every single neuron is just going to be a weighted sum of a bunch of inputs coming in from V2 that are pooled and rectified. Now, how do you fit this? You fit it by using basically leveraging the modern neural network backpropagation fitting algorithms that have been developed. So we could not have fit this, 30 years ago, but now there's a lot of software. Current thing is PyTorch. This was actually written in an older piece of software called Keras, for those of you who care, that basically allows us to fit very complicated series of stages to just output responses. That's how all modern neural networks are trained. Just for completeness, we also compared this to a standard convolutional neural network, which is VGG. I think this comparison is kind of unfair because VGG is trained on natural images, and these data come from movies. But that's the current sort of figure of merit right now. Nobody has a neural network model of the visual system in primates or humans that uses movies. They are all trained on static images. So we have movie data. We want to model the whole space-time response of this system. And so we're just comparing to the best model out there. This model, all the way to V4, from the image to V4, requires about 20,000 parameters. So if I did this in a convolutional neural network, again, I would have millions of parameters. This is a very low-dimensional model. So how well does it work? It works quite well. So on the upper left here, you can see a parastimulus time histogram. The black is the neural response over time, and the red is the predictions of the model. And you can see that the model predicts really, really well. On the upper right here is, again, this CC norm metric, which shows basically how well the model is predicting neural responses. Notice the model of V4 does not predict responses as well as the model of V1 did. The model of V1 was predicting up at 0.8 or 0.9 correlation, and here we only have about a 0.5 correlation. Now, you might say, oh, that's horrible, but remember, prior to this, the correlation between models and responses in V4 was, even for static images, not movies, was less than 0.2. And I should point out one other thing. For 40 years, the best V1 models we had, which everybody believed, and I still believe, which are these Gabor wavelet V1 models, they predicted with a CC norm of about 0.5. So the way to think about this is our current model, this hierarchical convolutional energy model of V4, is basically as good as the V1 models we all going around believing in for 50 years. There's some other things on this slide that aren't important. Just two things I'll mention briefly. One is whenever you're fitting complicated models, it's not only important to have stimuli be separate fit and test data sets. You have to have your subjects be separate fit and test data sets. And the reason for that is if you fit to all your subjects, you can actually overfit the model framework. So in this experiment, what we did is we only used one of the animals to develop all the modeling framework for this project. And we never looked at the data from the second animal. And then after the model was frozen, and the only thing we're going to do is fit the free parameters of the model to the data, we unmask the second animal and analyze that. And you can see that the two animals are virtually identical. So this model is not overfit to the animals. And the last thing I will mention is down here on the lower right, this shows model performance versus the amount of data. We have like 100 times more data than anybody else, and you can see that the models are still improving. So even at 1 million frames of video, it's not enough to really fit these models really well. We need even more data. I doubt that will ever happen, but it would be nice to have. All right, so now we got a problem. Great, I have this model of V4. It seems to work really well. What does it do? How do the receptive fields look? What is it actually coding? It's some gobbledygook about having a V1 feeding into another V1 feeding into another V1. But what does that really mean intuitively? What are the features that this thing is representing? And that's the fun part of a lot of neuroscience, which is the interpretation phase that you do after you fit your models. This interpretation phase, in this particular case, is complicated because these models have 20,000 parameters and they're highly nonlinear. So we developed this method basically building on a lot of things people have done before, basically trying to create what we call the predicted optimal stimulus. This is essentially, you run your neural network and force it to try to generate as high an output as possible. And that's kind of that's your prediction about what the best possible pattern is that would stimulate this cell. Now, when you do this, essentially what you're doing is you're feeding in random data to your neural network. Just remember, our neural network here is the model fit to one V4 neuron, right? We have 313 neurons altogether. We're going to do this separately for each individual neuron. So now I'm going to feed random noise into my neural network, and I'm going to do gradient descent to optimize the response of this neural network. And that will kind of like how a diffusion model works in the neural network field, that will cause the unstructured random noise to converge on some image. And that image is the most likely, the image that best evokes a response from the neuron. There's a problem with doing this, which is that it could potentially depend on the random conditions that you initialize. So you can't do this just once, you have to do this thousands of times. You start with random noise, you do gradient descent, you get a pattern. Start with random noise, you do gradient descent, you get a different pattern. And then you compare all those patterns to one another to see how similar they are. And we did all of that in a way that you don't want to know about in this talk. Just trust me, we did it right. And what we find is about 80 to 90% of the neurons just have one mode. In other words, if you keep running this randomization over and over again, the predicted optimal pattern for the neuron is always very, very consistent. The phase will change because these are all complex cells, but the basic structure is the same by any variety of any metric that doesn't take into account phase. About 10% of the neurons have more than one peak. So there's two different modes, and depending on where you start, you might end up with one predicted optimal pattern versus another predicted optimal pattern. But this is not a big effect. It's a modest effect, even in the 20% of cells that have more than one peak. So at this point, let's just declare victory and assume everything's unimodal and continue on. This means we can actually interpret the predicted optimal patterns that we have for all our 313 neurons. So here's some V4 receptive fields. And remember, these are from movies. So these receptive fields, unlike most of the receptive fields you may have seen, live in space and time. And this is just 4 neurons. You can see the prediction performance on the bottom. These are 4 decent neurons. And you can also see at this point, these things are really weird and cool and complicated. Let's look at the one on the left. This looks like two oriented low frequency, a low frequency oriented green grading tilted to the right, and maybe a vertical blue-yellow grading. But the oriented green grading also has a grading on top of it that seems to be like a high frequency left diagonal grading. And this receptive field structure is essentially a receptive field structure that you would get by building a cell that is concerned with amplitude modulation. It's essentially multiplying together other gabors to create a new kind of receptive field. Unless you're like an engineer, you might wonder, what in the hell is amplitude modulation? Why do I care? I'm just going to tell you, if you're trying to build a visual system that can detect, say, illusory contours, or that can break camouflage, it turns out building that system out of amplitude modulated neurons is a really good thing to do. And we think this has to do with camouflage breaking. Oops, it stopped. Why did it stop? Here's another one. The second receptive field here, you see this is a higher frequency vertical grading modulated by a low frequency and time shifting color grading. This is again an AM grading, selective cell. The cell on the third panel seems to be more complicated. It seems to be changing its tuning over time. Its early tuning is different than its late tuning. And the cell on the right, what in the heck is going on with that? It just looks like noisy shmoo, but this cell is actually pretty well predicted. So this seems to be a texture cell. And in fact, turns out that V4 neurons, if you think about it, like what is the first principal component of the receptive fields in V4, contour versus texture is actually the first PC. So this is one of the texture cells we see in V4. Okay. Another game you can play is to actually take this predicted optimal pattern, which we got by fitting our V4 model and then generating random noise and then optimizing that noise to find what the predicted optimal pattern is. We also have the data from these neurons where we showed them, several 100,000 to 1 million frames of video. And we can look through that data to find the actual pattern that elicited the largest response from the cell. And we can look to see if how the correspondence, try that again. We can evaluate the correspondence between the predicted optimal pattern and the actual pattern that elicited a response from the cell to see if those correspond. This is kind of a sanity check. So you can see on the bottom here, the best response for this neuron was a movie of a little penguin. And you can see that the predicted optimal pattern that best matches the algorithm tells us is the most effective stimulus for the cell, matches the wing of this penguin. This is one of my favorite slides. This shows the original frame on the left for it's like 10 different neurons, the predicted optimal pattern on the right, and the union of them, meaning the overlay of the predicted optimal pattern on the original frame. So if you look at the second row on the left here, this thing is called a prairie chicken. We don't have them in California. This is like somewhere in the Midwest. And you can see this is a pretty complicated little animal. You can see the predicted optimal pattern is this kind of herringbone structure. But if you overlay the predicted optimal pattern with the stimulus, with the frame that was the most effective frame for this V4 neuron, you can see that it matches perfectly the chestal area of this bird. So this random V4 neuron just sitting around there happens to be a chicken detector. It probably detects other things as well, but in the million stimuli we showed, the thing that matched the most was this prairie chicken. And you can see for all of these neurons, the responses are reasonable. Here's the penguin thing I showed you before. This is a toad. You can see that the predicted optimal pattern for this toad is these kind of curved green, red stimuli, and those, when you overlay them on the toad, do a really good job. Here's a uglier situation. This is, what is this? Oh, it's a Komodo dragon. All right. These were nature films. Okay. So there's a Komodo dragon. You can see the predicted optimal pattern on the right is some hideously complicated, who the heck knows what this thing is doing. But when you fit it over on the Komodo dragon, it actually matches features in the Komodo dragon. And that's true for all of these things. These predicted optimal patterns actually do match fairly well the most effective stimulus in the stimulus set. And the better the model is at predicting neural responses, the better the match is between the predicted optimal pattern and the most effective stimuli, which is what you'd expect if your model's working. So this tells me that basically this V4 model is a good model and it's producing accurate receptive fields, and I don't know what the heck they are. So, all right, so in neurophysiology is like a butterfly click. I have 313 neurons, and now I have receptive fields for all of these 313 neurons. What do I do with that? What neurophysiologists always want to do is they want to group things. Like, are there subclasses of neurons that do a specific job? And there's a good reason for doing this. If you played this game in V1, you'd end up with oriented achromatic cells and unoriented color cells, because there are two giant channels in V1, a chromatic channel and an achromatic channel. So the idea is if you went into V2, you'd end up with 3 groups of cells for the thick stripes, the thin stripes, and the inner stripes that all have different selectivity. You go into V4, we know there are giant color, they're known as color globs in V4. There are giant color domains, much bigger than the blobs in V1. But those color globs also have spatial information in them. The channels are mixed. And the interglobs in V4 also have some residual color selectivity. So the division between color and space in V4, we know just from other work, is much lower. So the question is, can we make functional clusters of cells in V4? And you can kind of eyeball this, and if you do that, you get kind of the following 4 clusters. You get some contour cells. These are almost always amplitude modulated cells. You get a bunch of curvature cells. The curvature cells make me very happy because my first big paper that got me a job was saying that V4 is tuned for curvature. So here's a completely independent way to show that same effect. There's not a lot of cells, but there are cells that show temporal contrast, meaning that the initial response of the V4 neuron to a movie is different than the later response inside the temporal response envelope of the cell. So they change their tuning over time. And then there are a bunch of neurons that just seem to be doing amplitude modulation in some complicated way. So that's fine. Eyeballing things is good to get a first cut, but if you're going to publish a paper, you have to do quantification. So the problem we have now is we have a hideously complicated 20,000 parameter model, and we have these predicted optimal patterns that are also hideously complicated. So what do? Our choice in this particular situation was to take the predicted optimal pattern and just focus on that and try to essentially classify those predicted optimal patterns objectively and quantitatively rather than just eyeballing. And the way we did this is we just took these pops and we moved them into various embeddings. So we had several spatial embeddings. We had which just means a nonlinear transform into some subspace, right? So we analyzed the pixels, the distribution of pixels. We took the power spectrum of the images and analyzed the power spectrum of the images. We had several other metrics that just focus on the spatial statistics of the pops. We actually separated the analysis into 3 parts. One focused on space, and one focused on color, and one focused on time, because we have all that data for all these neurons. And if you do this, you find out that you need about 4 principal components to account for the spectral distribution of these receptive fields. And you can see that these 4 principal components kind of delineate a space that where we've got high frequency versus low frequency neurons, radial versus curved Gabor's, 2 different kinds of curvature and principal component 3, who the heck knows what's going on there, and then low frequency neurons versus curved neurons on the 4th principal component. You can basically take this principal component solution for each of the metrics. Here we use several different metrics, and you can aggregate those together in order to get a classification, an objective classification of these 4 neurons rather than eyeball classification. And in that case, what you see is low frequency neurons, medium frequency neurons, high frequency kind of stripes, localized edges, these really hideously complicated dot texture neurons. You might think this just looks like noise, these can't be real receptive fields. No, these receptive fields down here on the second to bottom row predict just as well as the other receptive fields. These are real receptive fields. And then these stripy texture neurons. Of course, naming these is arbitrary, right? You can name them however you want. Naming isn't anything about the data. It's about thinking about the data. It's just a human limitation. You need to name things. Okay, so this is the spatial information in the receptive fields. We also analyze the color information in the receptive fields. Now, I don't know if anybody here is a color person, but any actual color psychophysicists will really find this objectionable. So please just go make coffee for a few minutes and then come back when we're done with the color. We didn't use a standard color model. The monitor we had was color calibrated, so that the colors were veridical, but it wasn't really psychometrically calibrated very well. So we just decided to stick with the RGB channels on the monitor. Those RGB channels are related to how human vision, color vision works, but they're definitely a poor model of human color vision. We mixed the RGB channels in a novel color space that we invented and basically recovered the principal components of the space. And so in that case, you get black-white neurons versus blue-yellow neurons, blue-yellow versus green-magenta, and red-cyan versus blue-yellow. And now you can look at the population distribution of these neurons as well. And so the main axis here is high frequency luminance versus low frequency color dominated neurons. And this is interesting because this is, of course, the same distinction I told you about several times that happens in V1. So although the color channels and the luminance channels are intermixed in V4, there's still, I would say, a gradient between these 2 extremes of color selectivity, low frequency, and spatial selectivity, high frequency. And you can also look at time. This is nice because almost nobody uses movies in studies of V4, so nobody looks at time in V4. Remember, I told you at the beginning that everybody thinks of vision as being a what pathway and a where pathway. And so in the what pathway, everybody ignores time, and in the where pathway, everybody ignores space. I'm using everybody loosely here, but largely, most of the where pathway studies, like say visual area MT, a very important motion area, or MST, the area that MT feeds into, those oftentimes use dots that have temporal structure but no spatial structure. And then studies of the object pathway down in the temporal lobe, most of those studies use static images. So this kind of gave us a rare opportunity to actually understand the temporal tuning of V4, an attribute that nobody has ever cared about before. But we had the data, so we did it. We took the pops, we looked at their temporal profiles, we did PCA on the temporal profiles. And when you do this, you end up with three principal components: tonic versus phasic neurons. That's a classic distinction that appears almost throughout the entire visual system, even as early as the LTN. Medium lag phasic neurons versus short lag phasic neurons. That's just an issue of how long it takes them to respond. And then medium lag phasic neurons versus tonic neurons. And then you can look at the temporal response profiles if you care. The blue here is the temporal responses for one animal. The red is temporal response of another animal. Notice there's a slight delay. One of these animals is older than the other and larger than the other. And so that animal's V4 is delayed relative to the other animal. But other than that, the temporal tuning is. So you can aggregate all this data. Oh, sorry. One other issue you can look at is, are these neurons space, color, time separable? So separability, those of you who don't dabble in engineering, separability just means, can I consider these dimensions on their own, or do I have to consider interactions between the dimensions? So obviously the system's going to be simpler and our lives are going to be simpler if space and color are separable. And the neurons are either, that basically you can consider the color tuning of a neuron without thinking about space and space tuning without thinking about color. Turns out that the neurons are largely separable, but not entirely separable. And I think space-time, they're fairly separable. Color time, they're less separable. So it seems like the color tuning is changing over time more frequently than the spatial tuning is changing. We're not exactly sure why. One thing to remember about color vision is it's slower than spatial vision. Color vision is slower and more low frequency than space. In your brain, your peak spatial frequency that you can process is about 60 cycles per degree, and the peak color spatial frequency you can process is, as I recall, about 12 cycles per degree. So color is a very low spatial bandwidth signal. Now, if you look back through the literature, it turns out there is a really old model from 2003 from a friend of mine, Mike Willicke, and Jan Karklin, who built a V4 model, purely theoretical. This had no physiology data in it at all. They basically made an artificial V4 model, and they showed what the receptive fields look like. And this V4 model basically is a sparse coding model. So remember I mentioned several times before that the idea of coding in V1 is that you have this Gabor wavelet pyramid, and that these Gabor wavelets are essentially trying to match the independent components of natural scenes, and that gives you a sparse representation. So Jan and Mike iterated this sparse V1 model, and they built a V2 model, and they built I think there was only one stage. So they took the V1 model and they put it into another sparse model, essentially asking the same question we did, which is, can you basically build a visual system by just having V1 on top of V1? And they get receptive fields that look like on the left here in their completely artificial model with no physiology data whatsoever in 2003. And if you notice, these receptive fields actually kind of look like our receptive fields, which I think is kind of cool. I've always loved this paper, and it was kind of neat to see that they weren't crazy. There's other ways you can think about what V4 is doing. One way to think about the Gabor wavelet representation in V1 is that it's taking derivatives of the image. And then if V2 is also taking derivatives of its input, then it's taking derivatives of derivatives. And then V4 is taking derivatives of derivatives of derivatives. If you're into derivatives, that's a cool way to think about it. I'm not, it confuses me, but my graduate student Mike loves this model. And another way you can think about it is that it's performing something called a scattering transform. A scattering transform is essentially a really cool machine learning tool for coming up with essentially an optimal random projection in a very, very high dimensional space. And of course, we always like it in vision if the model we're fitting is optimal, because we assume that evolution is trying to optimize everything it can, given the stuff that it has to work with, right? Evolution is a local minimum, but within that local minimum, it should try to be as optimal as it can. And because oftentimes I get a question of this in the questions. And this is my last slide. I just want to mention that there's one sort of elephant in the room that we're not talking about in this talk and in this model, and that's attention. Turns out attention, visual selective attention to either features or space, has about a 15 to 20% effect on V4 neurons. And this model is not an attention model. It does not have attention in it. We were not controlling attention in our subjects. So there are some amount of the variance in the V4 neuron we cannot account for, which is about 20% of the variance. So really our noise ceiling of what we could actually build in this experiment is actually 20% lower than the theoretical noise ceiling. So that's about it. I told you the HCE model, hierarchical convolutional energy model, implements a hierarchically organized repeating scheme of simple and complex filtering operations. This model provides good predictions of spike rates in V1, V2, and V4 under natural conditions, movies, and at high temporal resolution. Where this model, remember, is being I should take a step back. Oftentimes in physiology, people use static images. And they'll show an image, and then they'll build a model that predicts the neuron's response, say, average over 200 milliseconds. I'm not doing that here. We're predicting the response of this neuron to every single frame of the video. So the model is actually at 60 hertz. So we're predicting at high temporal resolution. V4 neurons are distributed across a 4 principal component spectral space, whose axes reflect spatial frequency, curvature, and texture. They're distributed across the 3 PC color space. Monochrome, blue, yellow, and green, magenta, and red cyan. These are actually the color axes you would expect based on everything we know about psychophysics and everything we know about how V1 codes information. V4 neurons have a broad distribution of different basic and tonic tuning properties. Most importantly, the tuning properties in V4 are not human interpretable. So I and every other neurophysiologist I know kind of assume at some point we would find the features that are encoded in V4, and you could look at them and say, oh, yeah, that makes sense. I understand what that is. It's like a little piece of curvature. No, that's not the way to think about V4. V4 is just coding complicated constellation of combinations of random V1 basis functions in this really hideously complicated way where functions are multiplied together in strange, unexpected ways because it's just trying to span the space of intermediate visual features. And oftentimes in V4 we think, well, there should be a neuron that's tuned like just for my face. There are neurons downstream that are tuned for faces, but V4 actually doesn't even really care about image segmentation. So you could imagine a V4 neuron that would be optimally tuned for halfway through my face, down to my shoulder, up, and including half the background. And that would be the optimal pattern for a V4 neuron. V4 neurons are just tiling the visual field. And the amount of attentional modulation is fairly low, and the figure ground segmentation is, for the most part, being booted downstream. What's happening in V4 is that illusory contours and contours in camouflage are being made explicit so that essentially you get a more complete image of the texture, a more complete representation of the texture and the contouring. And that's it. Thank you very much for your time. I hope that was interesting. I'll go back here. Questions?
Yes. So I'll start. How many neurons are there in V4? And did you try to encompass all of them, or are you taking a subset of them?
Oh God, how many neurons are there in V1? Well, V1 is 10% of your entire cerebral cortex. So if you have 20 billion neurons in your cerebral cortex, you've got 2 billion neurons in V1. V4's, about 20-25% as big as V1. So that probably means there's what? 1/5 of 2 billion. There's like a couple 100 million neurons. We've got 300. Welcome to neurophysiology. One of the almost everything in neuroscience, for those of you who aren't neuroscientists, almost everything in neuroscience is measurement, right? In any area of science, there's 2 things that can block your progress. One is you can't measure stuff, and one is you can't model the stuff. And at any given point in any area of science, one of these 2 factors is limiting you. And in neuroscience, it's definitely measurement. So, all of these, we were in this experiment recording from maybe 5 to 10 neurons a day, and we had to record them from for months. The new neural pixels technology that everybody's starting to use now enables you to record from 1,000 neurons a day. So the data rate is increasing dramatically. I don't think that will have a big effect in V4 because V4 is a pretty dumb visual area. And I say that as a person who studied V4 all my life. But when you're thinking about prefrontal cortex, where we have no idea how information is coded or represented, how you divide prefrontal cortex into areas, something like that, then these really large scale recordings will be very, very valuable.
I was wondering whether you human psychophysics, the relationship between form coherence and discrimination, given that the children are seeing the model that they should be ideally related to, as well as the color damage in terms of iron which we can see. So have you considered what have people actually done, let's say, a correlational analysis where you see I don't know,
So the question is, what is the psychophysics relating color discrimination to form discrimination, and how does it relate to these data? Yeah. Well, I've got both good news and bad news on that front. There's a lot of neurophysiologically motivated work on form and color discrimination. And almost all of that work is motivated by what we know about form and color representation in the LGN, in the retina, in the LGN, and in V1, where we know a lot about how those two sort of domains of stimuli are represented. But there's been no work that's been motivated beyond that because there are essentially no studies that look at color form together outside of V1. So it's kind of terra incognita. One thing to remember is this is an intermediate stage of processing, V4, and there are probably 10 stages of processing, probably 20, to get from V4 to the neuron that you're recording from when you're doing psychophysics, which is the one attached to this finger here. So there's a lot of other neurotransformations that are going to occur between V4 and your finger response. So I tend to think of these, I think we were discussing this actually just before the talk. If you're a psychophysicist wanting to understand form and color information, the real power of psychophysics is you can ask questions about what information does not get through does not get into the system at all, and people can't make judgments about it. Right? And once the information gets into the system and it's kind of percolating along in memory circuits, then you're in the realm of perceptual psychology and things get a lot more complicated. So it's not obvious to me that we're going to be able to see a signature of this spatial form integration that you see in V4 unless we can identify essentially a bottleneck constraint. It's like, given these, the logic would be, given what I know about V4 now, there should be stimuli that say I can't discriminate. And if I make those stimuli, is it true that people can't discriminate? Because essentially there's a information loss in V4, right? And that would be a cool game to play, and nobody's done anything like that.
Amazing work as usual. So taking from the interpolation between V4 and psychophysics of behavior, do you think the HCE is propagated after V4? And the reason I ask that is because if you just keep building V1 after V1 after V1, you are only going to recognize object from one viewpoint and with a certain type of motion. So what's your thought on that?
That's an interesting question. So the question is it turtles all the way down? And my prior would be, yes, it's turtles all the way down, because what the heck, it works. You know, we have no model of how neurons, of how information is actually represented in infratemporal cortex. Those, we have a lot of studies of those neurons. They seem to be selective for individual objects, but the stimulus sets are fairly low, so they may be selected for a lot of different objects. We don't really know. It seems like there's some sort of columnar organization that may have to do with feature tuning for objects in infratemporal cortex. But the amount of data we have is fairly small. People or animals usually aren't doing an attention task in those studies. There are a lot of artificial constraints put on studies of IT that limit the amount that we know. Traditionally in neurophysiology, everybody wants a model that they can understand. And that means they want a model that has features that are interpretable. Turns out there's a way to build an infratemporal cortex that results only in uninterpretable features. There is a mathematical theorem called compressed sensing that basically says if you have a very high dimensional space that you want to encode, that there's no optimal way to encode it, and you just pick random basis vectors. And in that case, what that theorem says is that everybody's IT is different. All the neurons in IT are just coding random stuff, but because the space they're coding is natural images, which is a very high dimensional space, we all end up coding equally efficiently. So that perspective is kind of the nihilistic perspective, says there's no representation at all in IT. And the alternate perspective is something like this. Well, I mean, you could get there through this scheme too, just iterating, right? So I don't think anybody really knows at this point what the answer is going to be, like how far this model can be pushed and whether it will make any sense further on. Another thing I should mention, because I mentioned attention in the last slide, is attention scales with distance from the periphery. So the attentional modulation, if you have an animal or a human doing an attention task in peripheral visual cortex, and you're recording from neurons, the amount that the neurons are modulated is like 2%. It's so small that nobody's even sure if it's real or it's just some artifact of residual eye movements. In V4, it's about 8%. Excuse me, in V2, it's about 8%. In V4, it's about 15%. The next stage of processing in PIT, it's about, say, 30%. The next stage of processing after that, it's 50%, 60%. When you get to prefrontal cortex of humans or animals, because we've done studies in both now, neurons are basically 100% modulated by attention. And so those neurons can't be described just in terms of their stimulus selectivity. To describe what those neurons do, you need to think about the stimulus selectivity and the task together because they jointly determine tuning. And so one thing that will clearly happen if you take this V1 iterative scheme is as we go forward in the visual system, attention is going to be more important, and trying to understand how attention affects these representations is going to just complicate matters. I know that was a long answer, but it was really a long way of saying, I don't know. Any other questions?
Yeah, that actually partially answered my question. So my question was on this slide, and if it does or could take into account attention to improve upon it, because in my mind, each of these slides is basically where I would focus, because I'm assuming that that's where the movement is in that movie with these different animals. So where it gets worse is where my mind, as soon as I see like the Komodo dragon, maybe it's flicking its tongue and moving its arm. So my attention would move to multiple places and scan that. So I'm wondering kind of what you were just talking about. If, as you're building this model, if including more focus on the attention would improve that.
Okay, so if we're going to build a model of the visual system, even in V1, focusing on attention is going to improve the model just a tiny, tiny bit, right? In V4, it's going to improve our model by 20%. And if you remember, we were predicting about 80 or 90% of the noise ceiling in V1 and V2. We're only predicting 50% of the noise ceiling in V4. But if 20% of the variance is we can't account for because we didn't control attention, then really our ratio is not 50%, it's more like 70 or 80%. Right? So I think the reason this V4 model is not performing as well as V1 is largely precisely because of attention. For those of you who do psychophysics, you will know that measuring attention is hard. Generally, you have to instruct the person or the animal to attend, and that's annoying and hard. An animal doesn't want to attend where you want it to attend, so it's hard to train them. Humans oftentimes just don't do what you tell them to do. And you don't really have any objective measure of attention because attention, the way I think about attention, my own personal view is attention is essentially the control system that's controlling the representation of information throughout the entire brain. And so it's operating at all levels all the time. Attention is constantly modulating neural responses to optimize the system for the task at hand, which means increasing the representation of task-relevant features and decreasing the representation of task-irrelevant features. And attention also controls the way information flows from long-term memory into working memory, and it tries to essentially optimize the amount of long-term information relevant for the task, right? So it's operating everywhere, and what we would ideally like to have is really good attentional control, but that will slow up an experiment dramatically. Because we have, if you think about just two attention conditions, say I had attend to the foreground versus attend to the background in this experiment. Now, instead of 1 million frames of video, I need 2 million frames of video, right, for these two attention conditions. So adding attention on top of this just makes everything much, much harder. And that's the reason that attention, people study attention, and everybody else tries to ignore them as best they can. It's, you know, we're all just humans. We just want to try to get our job done.
Thank you for that really interesting talk. So I was kind of curious, has anyone tried to like correlate cellular heterogeneity within the retina to subtle differences within receptor fields? I'm thinking kind of like cone mosaicism, for example, between individuals. And does that heterogeneity get more pronounced in deeper parts of the visual cortex?
Well, I'm not sure which of two questions you're asking. So one possibility is you're asking about the heterogeneity of receptors or ganglion cells in V1 and how they feed into these neurons. And the other, you're talking about heterogeneity across people.
I was thinking, yeah, more so heterogeneity within specific cell types of the retina between individuals. For example, like, you know, they have different ratios of cone photoreceptors even before the retinal ganglion cells, and how that might, you know, influence
that's a
kind of
very
receptor field space within different
that's a very interesting and valuable and important question, because of course, the retina is your sensor layer, right? If your sensor changes, it's got to be changing all kinds of stuff downstream. And there is a large body of work, both psychophysically and in neurophysiology, trying to understand how changes in the sensors, changes in the photoreceptors or the ganglion cells affect downstream areas. And there's also a lot of basic work trying to understand if different types of neurons in V1 receive input from different kinds of sensors in the retina. And of course, you can kind of guess that they must. If you have a blob cell in V1 that's color selective and low spatial frequency, it's probably either getting information from different cells in the retina or aggregating that information to ganglion cells differently, right? So there's a lot of that work done, I would say, between V1 and LGN and, the retina and LGN and the retina and V1. After that, there's basically nothing.
Do you think your models could eventually account for that?
It would be really interesting. If you notice, I know a lot of people here work on the retina, and I'm sorry for offending you. There was no retina model. Our model started at V1. That is not necessary. We could have built a retina model and fit that retina model too. It's not a problem. So in theory, that could be done, and it would actually be a really cool thing to do. And the way you would do it is you take the same strategy. We know what the assortment of cells in the retina is. We know what the assortment of ganglion cells is. So those are just now populations of potential building blocks that I can use machine learning to optimize in my entire chain. I've been talking about this. This work's 10 years old. I've been talking about this 10 years. You're the first person who's never asked that. That's a really interesting idea. I like it. I don't know if anybody on my lab will do it, but we will release these data. And there's a really great online viewer, Tom Dupree-Latour built, that allows you to interrogate these data and interact with it. That's one thing that my lab does a lot of. And so these data will be available on that would be a great project for somebody. Another question?
Oh, yeah. Thank you for that talk. I actually had a question about this slide. So I found it kind of interesting that a lot of the pops shown seem to map to animals. I don't know if that was intentional with your choice, like which ones you wanted to show. But I was wondering if it's maybe mapping toward objects or animals, right, in some way. And would you see that if you took the pops of lower or higher layers in this hierarchy? Like you look at just V1, first V1. What are the pops?
So, okay, so let me answer the second question first. Okay. I think if you took pops from V1, your intuition is correct, that they would be more scattered and they would fit more random features in the image. Because V1 is just representing little shape elements of edges, and it's not representing the camouflage edges, it's representing the high, it's going to respond preferentially to the high contrast edges. I think the reason that I hadn't really noticed that this slide is very animal biased, but I think there's a good reason for that, which is these are movies, not static images. And if you have a nature documentary, if the camera oftentimes is stationary or it's panning very, very slowly. There's one thing in nature documentary that's moving a lot, and that's going to be like plants in the wind or animals. And all of your visual system is designed to respond at a much higher rate to moving things than static things. And so I think what's happening is since we're looking at the peak rate of the neuron's response, it's almost always to something moving. And that's why we get animals in this. So in some sense, you can think of it as an artifact because, of course, these neurons have to respond to everything. And so, every one of these V4 neurons is going to respond to a static scene as well. It just won't be as high. And so one might argue, and maybe one direction you're going is, well, why don't you show me how it's responding to just static images, like exclude all the motion? And we could do that, but the thing to remember is the region of support for neurons for responses off the peak is much larger, right? So if you look at the peak response for a neuron in the visual system anywhere, there's only going to be a few stimuli that are going to elicit that peak response. But as you go down from the peak response to, say, a medium response, the range of neurons that will elicit a medium response is much, much, much larger, right? So that just makes it more complicated.
Thank you.
That was another, I should mention, I always love it when people ask me questions that nobody's asking for. Nobody's ever mentioned that. I've been showing this slide forever. So nobody's ever mentioned that before. Glad you noticed that.
Thank you.
Hi, thanks for the talk. I'm Marco, and I work for Philip Kaiser. We are pharmacologists. We are not neuroscientists. So if you can...
Well, look at the bright side. You can solve a problem. Neuroscience never.
If you can achieve what you want to achieve. So the final thing, what would be a device, an algorithm?
Well, this is basic science, right? So this has no usefulness at all. This is just understanding. So, there are 40 visual areas, probably 50 or 60 visual areas in humans. And essentially the goal of my lab is to understand representation in all these areas and how those representations are modulated by taught down by like learning and memory and attention. And so a lot of our vision work uses neurophysiology. Our MRI work uses MRI, but the question from my lab is always the same, which is what features are represented. So the way to think about this is, it's like a large language model. You think about the brain like a large language model or a convolutional network, even though those are both trivially simple kinds of architectures relative to the brain's networks. But, a lot of the questions people ask in the large language model community is, what are the different layers of my language model representing? What features are represented? And because a neural network is not obviously understandable. So you have to do something else to understand it. And so just like in AI, a lot of the work focuses on trying to understand what is being represented at each level of your network. Here in the brain, we're trying to understand what is being represented at each level of the network. Now, is that useful in the immediate future? No, it's not useful. You could, it has one use you can think of, okay. In the immediate future, it's not useful. Is it potentially useful for anybody, anywhere, anytime? And the answer would be, yeah, sure. If you understand how information is represented across this hierarchy, then you can decode it. You can build a better decoder. In fact, there's a symmetry between encoding and decoding because Bayes' theorem basically says if you can build an encoding model, which is what we have here, you can flip it on its head and turn it into a decoder. So the better our encoding model is, the better our decoder is. You can imagine now, if you can decode, now you can build a prosthetic. Right? So you can put electrodes in the brain and you can decode information from that brain. That's a use. Another use would be, say, bypassing a brain and sending something to a computer. So imagine somebody has a brain lesion. This is obviously future technology because we can't really record from the human brain very well right now. Somebody has a brain lesion in a very focal area, a bottleneck area. Most brain lesions that humans suffer, let me take a step back. The brain is a really complicated network. The cerebral cortex, just ignoring the cerebellum for now, cerebral cortex has got 20 billion neurons, and they're organized into probably 500 distinct brain areas. And each brain area has a 50% chance of being connected to every other brain area. And every one of these connections has feed forward and feedback and a collateral that goes to the thalamus. So it's this really hideously complicated network. If you have a brain lesion that's just kind of random in this network, probably, if it's a small lesion, it will make you slightly worse at everything, but you may not even notice it because there's a lot of other routes for the information to go around, right? If you have a brain lesion in a bottleneck area that has no alternate pathways, that lesion is going to be profound and identifiable. Like it will cause a certain specific deficit. Like just last week, we were scanning at Berkeley a person who was cognitively completely intact but couldn't speak because of a stroke. So in that case, you could imagine building a prosthetic that just bypasses that area. Okay, I have input from the areas that feed into it. I have output, the areas that go into it, that it's going to feed into, but I don't want that to be a straight shot because those areas, the output areas, are expecting that it has this information that's represented. And this information that's represented is implicit in the inputs, but it's only explicitly represented in the stage. So if I have a decoder, I could build a bypass circuit that would go around. So that's the way to think about this. Now, this is all like fantasy. For those of you who worry, is my brain going to be decoded in the short term? The answer is no. We have no technology for decoding brains right now. The two best technologies for decoding brains are ECOG, and of course you would notice if you had ECOG electrodes inserted in your brain, it involves a very large, nasty surgery, and MRI. But you also notice if you're an MRI machine. So nobody's going to have their brain decoded like remotely, using space lasers or some other weird technology. That stuff does not exist, and it will probably never exist.
Hi, Dr. Gallant. I'm Dominic Lewandowski, Philip Kaiser Lab. I will probably ask the last question. And thank you so much for a great talk and great discussion. My question would be more practical, I think. When we evaluate quality of vision in retina disease models, and we, for example, measure visually evoked potentials in V1, right? In a disease versus a normal control with healthy conditions. And what do you think? Because we evaluating V1, but looking at the high contrast, like drifting gratings and stuff like that. But to assess the quality of vision using moving objects, like colorful, normal everyday life objects, not just a drifting black and white drifting gratings. What part of the visual cortex would be the best to check those responses? V1, V2, V4, or maybe some other part?
Well, since you're doing visually evoked potentials, V1 is like the golden place to do that, because V1 has the highest temporal sensitivity of pretty much any visual area. And it responds very phasically to either a stimulus onset transient or to an eye movement. So every time you make an eye movement during natural vision, you get this giant volley of responses in V1. For those of you who are old like me, you may remember there were these old amplifiers called a lock-in amplifier that basically, if you have a non-stationary signal, the amplifier will lock into one mode of that signal, and then the minute the signal changes, the amplifier will essentially reset and lock into the new signal. And you can think of V1 as doing that. Every information comes up from the eye, it goes into V1. There's been a transient, so all the neurons in V1 kind of respond phasically. That phasic response kind of gets sent upstream and tells the whole visual system, okay, reset now, because I just made an eye movement. And then that's what you're going to see in a visually evoked potential. That's going to be modulated by the stimulus. The minute you get out of V1, the temporal response properties of the neurons gets longer and longer and longer because they're integrating over time. And so you don't end up with a good phasic response, and everything just gets harder. So I would say, stick with V1. The question is, okay, if you're going to do an assessment in V1, should you use gratings or should you use natural images? And frankly, if you were asking about V4, I would say use natural images, because any stimulus you use cannot, other than natural images, cannot span enough of the diversity of neural response profiles in V4 to make it worth your while. But V1 is really dumb as a bag of rocks. It's a bunch of Gabor-Wheeler filters. And we know that model works really, really well. It's like one of the biggest success stories in neuroscience is the model of V1. So I think in V1, using the stimuli that are optimized for that system, which is Gabor gratings, is a perfectly valid thing to do.
Thanks so much. Yeah, I think we have the next meeting coming soon, so I think we should finish unless somebody else has some. Yeah, we have to finish. Thank you so much.